Under the mentors of me, Tim Walston, and Dr. Scott Thatcher. And they're going to be talking about an automated scheme for image segmentation of the 4-cell C. elegans embryo.
As Dr. Walston said, I'm Kurt Warnhoff, and this is Allie Merman, and we are going to be we've been working under the guidance of Dr. Walston and Dr. Thatcher. And today we're going to be discussing our summer goal of creating an automated scheme for image segmentation for the 4-cell stage of Caenorhabditis elegans embryos. We're going to begin with a brief introduction on why C. elegans is a good model organism for our model and why our model fits the organism. And then we're going to move into where the project stands currently with past summer's efforts and where we hope to take it within this summer. We're going to be ready this summer. So first we're going to do a discussion on why C. elegans is right for us. C. elegans is a 1 millimeter long nematode that exhibits several very interesting and important characteristics that allow us to use it as a great model and coincides very well with our project. C. elegans has a very rapid period of embryogenesis. Within 16 hours, we can go from a 1-cell fertilized egg to a hatched young nematode worm. And this is important because within our project, we're looking at modeling embryogenesis, and this quick sort of embryological process allows us to efficiently gather data that we can use and segment and put into our model. If it was, say, like a 4 or 5 day or even possibly a month embryogenesis, it would not be feasible really for us to gather data on that and with the quality that we're looking at. Secondly, C. elegans is very easy to maintain in large quantities. Within a single petri dish, you can maintain a large population of worms, and creating populations is as simple as transferring several worms and allowing several days for the worms to mature. Also, obtaining the embryos, which are not fully visible there, but the embryos are visible to the transparent cuticle, and it's a fairly simple process to do. It's easy. With a couple of days' work, you can turn it out pretty quickly. So obtaining the embryos that we need for the image segmentation is a relatively easy process. Thirdly, the predominant adult form of C. elegans is hermaphroditic, and even more importantly than that, it's capable of self-fertilization. And this just basically means that we're able to generate offspring without having to swap gametes with another worm. And this just, again, adds to the efficient and rapid process of reproduction and embryogenesis, which is important for our model. Perhaps one of the most important aspects of C. elegans is that the eggshell of the embryos is transparent, and this really allows for our model to work for what we hope to do. We're able to see the shifts and orientations within the embryo, and that allows us to take video and capture these changes and model using image segmentation techniques that I'll talk about in a little bit. But if we didn't have this transparent eggshell, we wouldn't be able to do any of the work that we're doing without further applications. Continuing with C. elegans. C. elegans has an invariant cell lineage that gives rise to a relatively small number of somatic cells, 959. And the fact that cell lineage is invariant, is very important. And what it means basically is if you took 2 single-cell embryos and observed them side by side, the divisions and orientations of each cell would be identical between the two. So if we manage to make a model that gathers several data sets, we would be able to apply it across the entire species and look at changes within the normal type, so we can apply it to possibly applying to mutants and seeing what changes are different and what forces possibly affecting in a different way. And finally, genetically, C. elegans has a relatively compact genome. As you can see, comparatively to humans, humans have roughly 23 to 2,400 genes, while C. elegans about 20,000. And this is excluding phenomena like alternative splicing and other more complex mechanisms that humans possess, but suffice to say that genes are relatively the same, and when you compare genome size, C. elegans has roughly 3% of the genome that humans have. So the genes are much more tightly packed than C. elegans genome and this is useful again for hopefully further studies with genomics of mutants. For now, we're going to talk about our actual model we're looking to model within the 4-cell stage. This slide is really important in that it shows time shots within the 4-cell stage of C. elegans embryos, which is what we're looking at. Clearly, EMS from 0 minutes to 5 minutes, 7, 13 undergoes some very interesting shape changes. It elongates, and then elongates further, and then comes back to sort of the traditional shape of the cell that we see here. And what our model is hoping to do, we're trying to input parameters that will model this behavior, and hopefully by observing our model and looking at the code, we can say something about the forces that are actually acting upon each individual plasmid within the stage in the cell. And by doing this, we can hopefully gain an understanding of the intracellular forces that are acting upon this system. So, up to this point, we've had a summer's work. Last summer, Clayton Davis and Elise Wall put a lot of work into this project, and they accomplished some great things. Last summer, they managed to manually segment a 4-dimensional microscopic data set, and this is really, it's just one bullet point, but it's not a small feat at all, as Alex can talk about later. But 4-dimensional microscopy essentially gives you dimensions of length and width in different time stacks, which act as depth and then over a period of time. So for any individual time step, you can have up to 18 to 20 focal planes that would eventually lead to several hundred images that need to be segmented. And manually segmenting these images is a pretty tedious process, but I'm not going to get into that now because that will be covered later. But after segmenting these images, or the actual images on a microscope, they input an image to this GGH model that incorporates several different parameters down here, such as target volume, curved surface area, cell adhesion, cell age plus lying forces. The code for central movement is not necessarily a parameter, it's more independent. It's an independent code that models centrosomes within each individual cell. And the way these centrosomes are modeled right now, it's sort of a distance thing. The centrosomes are told to move as far apart within the cell boundaries as they possibly can. And as I'll get into later, this isn't really what we need, but it does incorporate the centrosome, so it's a good step in the right direction so far. So, while we have a lot of great things to work on, we definitely have some things we want to incorporate this summer and incorporate it into the model. Currently, calculations for this project and the model rely on a single set of data, which makes sense because it's so hard to acquire data. But well, if we want to justify our model as a usable biological model, we really need to collect a lot more sets of data to implement into this code. Without it, it's really hard to say. With only one set of data, you're much more prone to error within our model. And by getting more trials, I would feel safer about saying our model is actually doing what the biological system does. So basically what it breaks down to is the more data we have, the more reliable our model becomes. Talking about the centrosome movement, right now, as I mentioned, the code just makes the centrosome move as far apart as possible. However, what we need to do is model the centrosomes so that they follow a biologically significant pathway. And Ali's going to go ahead and this video, and I'm going to talk about it as it goes. This is an image of a single cell, well, 2 pronuclei, the union of 2 pronuclei, sperm and egg, and the initial division of this embryo. And here we didn't use this. This was taken from the site that's on the website, on the slide. But there's beta tubulin labeled GFP that labels the microtubules, and it allows us to really visualize these centrosomes, or I suppose more correctly, the microtubule organizing centers. And clearly, as you see the division occurring, they play a very important role in mitosis. They don't simply migrate away from each other. Their movements are directed and very important to the invariant lineage that we're talking about. So, if we ever plan on expanding this project into further embryogenesis or further mitotic divisions, we need to apply this concept, which is basic in the 1-cell, 2-cell stage. The same sort of concept can be applied to the 4-cell stage, and that will hopefully lead us into setting ourselves up for further mitotic divisions within our model. So here we are with two things we've talked about so far. And thirdly, we're going to talk about currently the model uses somewhat arbitrary values for target volume and surface area. And what we would like to do to hopefully add a little more biological foundation to the model is to segment a shellless embryo, which would accomplish the goal of allowing us to visualize where blasphemy would go if they were unconstrained by an eggshell. Therefore, the maximum and sort of ideal volume surface area could be reached, and we could segment that data and input it into the model, which would hopefully allow us to further validate our model as a biological system. But the problem with this is generating a shellless embryo is much more difficult than just isolating an embryo, and the procedure is very difficult procedure, and we are looking at trying to attempt it, but another possibility with this is trying to acquire a video from some other source and then segmenting that ourselves. So these are the problems we talked about, or not the problems, these are the areas we look to improve on in the summer, and we have one more that we're going to talk about, and that's the idea of improving image segmentation jobs.
So data is a huge portion of this project for us. We can't really move forward without getting more data in here because we're going to be continuing on online. Okay, so the first thing we're going to talk about is what is segmentation. A lot of you are computer scientists and maybe you're confused when we say segmentation. So segmentation is simply the process of partitioning a digital image into disjoint, non-overlapping regions. So we've got a picture of 4 cells, and we want to outline each cell in the eggshell and make sure that there's no overlapping the pictures as well. So currently, manual segmentation has been done on the entire data set. It is really inconsistent, it's really tedious to do, it's really boring to do. I'm sure MathBio doesn't want to pay us to playing games all day, they'd rather have us do something more productive. And the results from segmentation are really important for getting valid numbers into this model. If we get kind of crummy segmentation techniques, then we're going to get crummy values for surface area and volume, and even things that are in the images like pixel intensity that we might look at later on. So more limits of manual segmentation. Currently deploying DIM is a busier first tool, and I don't know how many of you ever played with this. I personally cannot get the hang of it. It's really hard for me. Each image takes approximately 6 minutes to segment by hand. This is if you're getting really good at the tool and you're hardcore segmenting, no distractions. So taking focal planes 11 time steps in theory would take 20 hours to do. In actuality, it took at least about 3 weeks to do. Taking care of worms and other biological priorities that you had to attend to. So 3 weeks is a lot of time for one data set. We can't spend our whole summer doing this by hand, so we need to figure out another way to do this. So when we look at segmentation problems, you have to look at what images you need to segment, because every segmentation problem is different from each image. So O's group, they have very different images than we do. They have a different approach for taking it. So we have pictures of cells. They're pretty round, which is nice. It's good for segmentation. We like convex things. And they're individual units, so we don't want any overlap of the lines. Also, since we only are looking at the 4-cell stage right now, we don't have to worry about a lot of complexity. Even if we choose an algorithm that's kind of computer intense, it's probably okay because there's not a lot to look for. Also, we're generating our own data sets this summer. We don't have existing data sets that we want to use. This means that we can pay a lot of attention to adjusting contrast and lighting measurements so that we can get the best image possible for the algorithm. Also, in most of the focal planes, except for the ones on top and the ones on bottom, you can usually see the nucleus. This is helpful for certain segmentation techniques we're kind of starting out. So we'll talk about that later on. So the first technique I'm going to talk about is the most basic. It's called thresholding. And there are a few types of thresholding. There are global and adaptive. So global, which I'll mention first, is where you take an image like this grayscale image right here, and you find a range of brightness values from light, dark to bright, and usually used for histogram. And you would pick a threshold value on there and put it on there. So any pixels that were below that threshold level would be black, and any above that threshold level would be white. And so you can see with this, it's kind of a crude segmentation, but it kind of works. It separates out foreground and background. Adaptive thresholding looks at little bits of the image at a time, so maybe it'll split it up into a grid and focus on just a certain neighborhood of pixels, and we'll have different threshold values for that. So that's really useful and important. Using methods like that, we can use this as a pre-processing step pretty easily. It's computationally simple, so we don't have to worry about it. No sweat there. The thresholding, as you can kind of see here, is not very effective for noisy images and just generally not clean images. Going to be the images we have is real data. So for example, up here, you can see kind of that there's this light spot here, and you can't really tell if that's data or if it's in the background, it's just lighting junk, and clearly this threshold value defines it as not important. So is it a good thing? Is it a bad thing? It's probably not okay, just let that go. Not think about that. So one other important thing about thresholding that is not too great is that there's no requirement for a continuous curve. And going back to things we really want to pay attention to when picking an algorithm is we want a continuous curve because we want our cell boundaries to be closed and continuous, because cells are closed and continuous. So that'd be great. So the first general class of segmentations and methods I'm going to talk about are contour-based. These are based off generally finding the boundary as opposed to the overall picture of the object. So most of these methods are the result of minimizing an energy function, which is kind of neat because GGH model is all about minimizing energy. Active contours are also called snakes, which is neat. And snakes involve a curve from initial seed or region to fit a boundary, basically within following the constraints of the function. So there are two main terms of this function, a data-driven term and a regularization term. The data-driven term basically just gets the curve to the actual boundary, it gets it sufficiently close. And the regularization term makes it smooth and nice. So data-driven terms have two classes. So kind of going back to the overall picture, these two main classes I'm talking about, contour-based methods versus region-based methods. Either of these can be implemented in a data-driven term. So right now we'll just focus on contour-based ones. Let's see. So contours bounds maximal edge evidence with minimal curvature to find curve length. And doing this encounters different problems. First and foremost, there's topological problems. It's often really prone to self-intersection, and that's not really good. A few other problems that we run into commonly with active contour-based methods are trying to decide when to stop evolving the curve. It's a parameter that the, programmer has to come up with when it's a good time to stop. If you don't match that correctly, then you'll miss your boundary and you have error there. Also, several algorithms have a hard time overcoming gaps between broken edges and selecting the initial contour. So, one active contour method that we're going to talk about specifically are level set methods. And level set methods avoid this topological problem, and specific level set algorithms, which there are many variations, address these gap problems in the initial contour setting. So, what are level sets? Anybody who's taking Comp 3, they know that you have a topographical or a three-dimensional curve, and you look on top of it, and you see the level sets on there, so that you can I'm going to talk about region-based methods. I don't have a lot of time to talk about general research-based methods. Bo's group is using a little bit of this. These also use global energy functions, but don't have to. So the difference between here is that they focus more on region, whole, and shape, as opposed to just line. So we're going to talk about a energy-free method called watershed segmentation. And this is an idea similar where you take a grayscale image, you look at the gradient of the image, basically the bright sections and the dark sections, and you consider that as a topographical map or surface, and you flood the minimum or the really low parts with water until you get to the top, and where the water would overflow or meet, you build watersheds. So you've got a nice clear line for segmentation. So I have a video, but I'm going to skip over it. I think it kind of gives me an idea here, but I'm sure there's examples. This is usually a very efficient method, and it will always yield continuous region. Although without planting seeds or markers to start flooding, you always get over segmentations problem, but it can be easily avoided. So seeds can be planted. So like I said, how we can usually see the nucleus of each image, we could use that potentially as a seed, or we could manually plant it. The problem with watershed segmentation is that they're difficult to smooth. The images are kind of noisy, little bouncy. It's not a great curve. To fix this, we use something called water snakes. Everybody should get really excited because water snakes take the watershed algorithm and add in ideas from level sets, mainly this idea of adding a energy equation, and so we have this regularization term that creates smooth boundaries. So here, this is a great example of this picture. It's got a 2-cell, which is kind of noisy, not that great. These are also fluorescentized. We don't have yet, maybe. They threshold it, and on the C right here, this is the just the watershed, plain old garden variety watershed algorithm. Which is close, but it's not great, as you can see. But after several iterations of this level set hybrid, then you see you get a much cleaner, much nicer approximation, which is something we really get. And so in conclusion, we talked about motivation, accomplishments, and all these different techniques.
You guys talked about an initial snake algorithm, I believe, which sounds similar but not identical to image seed carving. I wonder if you looked into that as a potential solution for your problem.
No. We had minimal time last semester to do research on this. When Clayton ran into this problem last year, where they were like, man, this is taking a long time to segment things. Shouldn't there be an easier way to do this? He found a paper on level sets and was like, hey, this is really neat. We could maybe use nucleus. So that was kind of our starting point for research, and found a lot of other regrowing methods kind of similar to that. So you might look into that. Might ask you about it later, because I'm really interested in learning all I can about the best segmentation techniques. This is also like eye candy of our model. So.
So, are you planning to go with this combination method for your segmentation?
I think so. We're going to do some more research. Picking an algorithm, I think, will be based off of one ease of implementation to some extent. Like, if it's something that I can't learn in 10 weeks, then that's not going to work. Also, accuracy is probably the most important one, because if it's not accurate, then it might as well be segmented by hand. And also, efficiency is kind of secondary.
We really need to just start generating data so that we have a better idea which of these segmentation techniques will work best for us.
Is that pretty much the goal for the summer then?
Yeah, it's kind of like the giant pre-processing step, so that next summer and fall we can really plow ahead with finishing the model.
I have a question. So you showed us all these different techniques, and you showed us how this one looks better than that one. Do you have a way other than this looks better than that one to actually automate the process of deciding how good of
In terms of like adding a number and saying this number's higher, so this picture's better.
Well, I mean, let me ask you another one. Other than looking at it and saying, this looks better than that one, do you have any way of quantifying how good one approach is to another with the results of these?
Not that I know of, but I think we're gonna kind of eye it now. Because I mean, like, for a lot of these techniques, for what we've been playing around with when we run these, it's so far off where you're like, that is wrong. Like, I don't need a number to tell me that it's wrong.
How about comparing it to the hand-done one from last summer? Could you take your different algorithms, see what you get versus what the hand-done one got? And,
We could definitely overlap and compare and contrast. One of the papers I was reading said that when like people draw boundaries, there's a bias to draw outside the line, like this huge bias in like how we draw. So it's kind of weird.
you might expect that to get exactly matching the
Yeah.
last year's one, but